All right, so the second half of gene expression is translation. So previously we did transcription, where we made an mRNA from the DNA. We modified that and produced a mature mRNA. So now we're going to do translation. So this is going to have 3 steps, and you're going to want to look at these figures as we go over this. We will have initiation, elongation, and then termination. So let's look at step 1. This is in figure 1114. In order for us to have this start, we're going to first have to have the mature mRNA find a ribosome. Now, as you can see in the picture, the mature mRNA is going to attach to the small part of the ribosome. So the mature mRNA binds to the small unit. So remember, this is when we're going to have several different pieces of our RNA coming together. So first we'll have the mature mRNA. So this is my mRNA. Then the small subunit, and that will attach. Then the first amino acid attaches with a tRNA. Now it turns out that the beginning of every sequence is going to start with the same 3 letters of the code. I'm not too worried about that now. Let's just get the general idea down. But remember, there are nucleotides that go along this mRNA. And remember, every 3 letters is a codon. So for instance, these 3 nucleotides are a codon. And the way the tRNA is going to be able to find the right place to sit is it's going to have an anticodon. So on the tRNA, remember this is that T-shaped thing. This is also an RNA molecule. So the ribosome is our RNA. The mRNA is our messenger mRNA, and here's our tRNA. And then on that tRNA, there's going to be an amino acid. So all 3 of these are going to have to come together, and then also, after that attaches, then the large subunit will attach on the other side. Well, the way the tRNA knows where to go, this is the anticodon. This is the codon on the mRNA, but the 3 letters that are with it that are complementary are the anticodon. So mRNA finds tRNA because codon will match anticodon. So let's take a look at that a little bit more. I'm not going to draw the ribosome because it just makes the picture a little bit blurry. But if this is my mRNA, my mRNA is going to have codons. So remember, every 3 letters of the mRNA is a codon. So here's the first codon. Here's the second codon. Well, the tRNA is also going to have 3 complements. So let's draw our tRNA for this one, and here it has an amino acid on it. So I'm just going to write it like that. So this tRNA, these 3 are the anticodon. And this is how they find each other. This is going to be an RNA, and this is going to be RNA, and they will have complementary base pairs. So for instance, if my mRNA has, let's say it has AAU, then the anticodon, is going to be complementary, and it is going to have UUA. Because remember, A and U are complementary in RNA. A and U are complementary. If the next three letters were CU or GUC, then the tRNA that would find it would have to be complementary to GUC. Opposite G is a C, opposite U is an A, opposite C is a G. So the anticodon on this next tRNA would be CAG. So the rules for how RNAs line up are going to be a little bit different than DNA. But if you have tRNA and it has to be complementary to the mRNA, then probably put the M here and the T here. But if there's an M in the mRNA, if there's an A, then opposite will be U in the tRNA. If there's a U in the mRNA, then opposite on the anticodon will be an A. C will be opposite G, and G will be opposite C. So this is going to be very important for our next step as well. So after initiation, after we bring together the ribosome, the first tRNA, and the mRNA, we're now going to begin to make our protein. It's going to begin to build. We only have one amino acid so far, but a protein is a series of amino acids. So I'm not going to look at all the different things. Let's just look at 5 codons that could become elongated. So here is my mRNA, and it has a series of nucleotides, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. So let's just look at 5 codons. So we can go ahead and we can start it with the same one that they have, AUG, CAU, U, G, 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 U, A, C, U, C. Let's just say that that's our mRNA message. We have 5 codons. So here's the first codon, here's the second codon, here's the third, here's the fourth, here's the fifth. During elongation, the only thing that happens is we're going to add more amino acids. So each time we'll have a tRNA come in. So it'll have an anticodon that will be UAC. And it'll bring over an amino acid. So we're not going to worry about which specific one. I know the book is showing specific ones, but let's just have an amino acid. So the first one comes in. We have an amino acid. Now the second one will come in. The anticodon opposite here would be GUA. And it'll bring over a second amino acid. The ribosome will then form a peptide bond between it. Well, now this tRNA, it's done its job. it can now leave and go find more amino acids to bring over. Now, we'll now have another tRNA come in. It will have an anticodon of ACC, because U goes to A, G goes to C, G goes to C. It'll have that anticodon. It'll have another amino acid attached to it. The ribosome will form a peptide bond. Well, now this tRNA has done its job. So now it can go off and it can go find another amino acid to bring to another thing. But as you can see, we're getting longer. That's why this is elongation. We now have 1, 2, 3 amino acids beginning. Another tRNA will come in. Opposite G is C, opposite U is A, opposite A is U. That's the anticodon that will bring over another amino acid. And we just keep building and building and building. So now our polypeptide, our protein, is now 4 amino acids long. Oops, forgot to put my peptide bond in there. So that's what elongation means. New tRNAs come in, match anticodon to codon. We form a peptide bond. We bring in another tRNA. Until finally we get to the last set. And there are what are called stop codons. These let us know we are done. So step 3 is termination. Once we get to a stop codon, we no longer have any more amino acids. Protein is finished. Even if there's more code, remember there's the poly A tail out there, we do not do anything with it. Once we get to the termination, we are done. So we have no more tRNAs to add. So here is our now completed protein. We have 4 amino acids, each with a peptide bond between them.